pathogens in urban stormwater. So the work that I'll be presenting on today is a combination of a whole heap of co-authors. So I've just put mine and Anna's name, but it really extends to a whole heap of people around Australia, and in fact some other people internationally as well. So the topic today is purely pathogens in stormwater. So I'm not covering treatment of pathogens in stormwater, just purely what's in stormwater. So a bit of an outline, so I'm going to the next slide now. A bit of an outline for today is that I'm going to talk a little bit about where we started with all this, which was back in 2006 in measuring fecal indicator organisms in urban stormwater, and whether or not we could predict their behaviour using a very simple conceptual model called MOPIS, Microorganism Prediction in Stormwater. And then we're going to go into some more details around pathogens in urban stormwater and a little bit about microbial source tracking, because I think source tracking is an important aspect when we're talking about pathogens in urban stormwater. So I'll get to details about that later. So on to the next slide now. So in the early years, as I said, we started with monitoring these fecal indicator organisms. So back in 2005 to 2007, we monitored about 4 catchments, ranging in event sizes, but mainly we've got around 20 events for each catchment, and around 14 events over each, 14 samples over each event, and analyse them for a range of indicator organisms. And we used that data to start looking at whether or not we could explain the variability of these organisms between events. So why does one event have particularly high levels of contamination versus another event at a particular site? And what we found was quite simply that there were antecedent climatic parameters such as vapor pressure, temperature, and relative humidities, which explained part of that variability between events, but also the rainfall intensities of the events at each site. So we found that 2 parameters were able to capture a significant proportion of that variability between different events. Some wash-off parameter, which was the rainfall, and some antecedent climatic parameter, which kind of represented the build-up of pollution on the catchment surface before the event started. We then looked at whether or not we could explain the within-event variability of E. coli, and whether or not some simple rainfall intensity metric could explain that variability. We weren't so happy with this, but it was as good as we got. We got in about 15% or 20% of the events, we got very significant correlations between rainfall intensities and E. coli, whereas in the majority of the events, they weren't significant. Anyway, we pressed on. This is the joy of modelling biologically active organisms in stormwater systems. So we developed this model called MOPIS. This is the little logo that we have here. So I'm just going with clicks now. So each click there will be another image come up. And MOPIS basically has 2 surface stores. So the next click now, there's a surface store and a subsurface store, basically simulating the build-up and die-off and wash-off of organisms in each of those surfaces or each of those stores. We tested this model on, so the next slide now, tested this model on a range of catchments. So in this particular slide you see we've got a prediction versus measured event mean concentration graph, and you can see that the Nash-Sutcliffe efficiency there was around 0.76, which is pretty good. And you see a table here with lots of numbers in it. Don't focus on all of the numbers, just focus on the top line there, which is called EC, which is the Nash-Sutcliffe efficiency for concentrations. And as you can see, for each of these sites, we're getting around 0.2 to around 0.4, 0.5 Nash-Sutcliffe efficiency. And to the untrained eye, some people might think that these numbers are quite low. For urban stormwater quality prediction, though, they're actually pretty good. In typical pollutants such as sediments, you're looking at actually less than that. So you're looking around 0.2. So these numbers are actually not too bad. If you just click one more time, you'll see another table pop up. We applied it to another 6 different catchments at a range of different scales, right down from Raleigh, which is a very small catchment, to the Yarra River catchment, which is a significantly large catchment in Melbourne. And you can see that the numbers there, again, are similar to what we saw before. So the model seems to work okay. Not perfectly, but pretty good for a non-conservative pollutant in stormwater. So I wouldn't say we conquered fecal indicator organisms in urban stormwater, but we wanted to move on and start looking at pathogens and seeing how we can use pathogens to better understand the risks of reusing stormwater. So we did a large collaborative grant with 4 states around Australia, and we monitored 10 different catchments with a range of different events and a range of different land uses in different climates. And we monitored these sites and we analysed them for a range of different constituents, and including pesticides, but today I'll just be focusing on the pathogens. You'll see a little symbol down here that looks like Coca-Cola. In fact, it's called the cave clan. The cave clan is a group of underground dwelling organisms. Unfortunately, the organisms are people. They hang around the caves of Melbourne, which basically are the stormwater drains of Melbourne. So during this project, we had a lot of our little sensors, which is this little blue thing you see next to the man standing there, ruined by, we thought, the cave clan, but they claim it wasn't there. So interesting. If you click to the next slide, you'll see a big table with lots of numbers. I'm not going to ask you to read all of those numbers, but mainly just the first row there, just to highlight the different pathogens that we analysed for. So we did a lot of different pathogens ranging from Campylobacter, adenoviruses, Legionella, we did some Staphy as well, Staphylococcus, and these numbers here just represent the percent positive samples in the catchments that we monitored. I'm not going to go into the details of this because I want to focus on one particular case study. So if you just click once more, you'll see a slide which is called Pathogens in Urban Stormwater Troups Creek Case Study. So this Troups Creek system is an urbanised catchment. The purple area you see on your slide at the moment is probably the only non-highly urbanised area. It's mainly hobby farms at the top end of the catchment. You'll see 2 blue, light blue shaded areas, one in the middle of the catchment and one down the bottom of the catchment. That's the wetlands, and we monitor the inlet of the downstream wetlands, so the one that's the bottom left-hand corner. So today I'll be just talking about the influent quality of that wetland to represent urban stormwater. So if you just click once more, you'll see 2 graphs, one with E. coli and one with Campylobacter. If you just click 4 more times, the 4 box plots will show up for you. So you can see on the left-hand side we've got E. coli concentrations. The open box plots are the ones that samples taken during dry weather. The closed box plots are those taken during wet weather. As you can see, E. coli concentrations are around what we typically find in urban stormwater, so around 10,000 per 10,000 to 100,000 per litre, or 10,000 per 100 mil. And wet weather concentrations are higher than dry weather, which is fairly standard. If you click once more, a grey box or grey shadow will come up over Campylobacter, the Campylobacter box plots. That grey shadow represents the range of Campylobacter that we typically find in Melbourne's wastewater systems. And so you can see the Campylobacter that we're measuring in Troups Creek, the stormwater drain, which would only contain stormwater, is having quite high levels of Campylobacter. So you can see median concentrations of greater than 100 Campylobacter per litre. This is culturable Campylobacter, by the way, not PCR. This is culture-based method. And so we're getting numbers up to around 1,000. We've got a couple of 1,500 up there, 1,500 per litre, which is quite high. So if you look into the Australian guidelines for stormwater harvesting and reuse, the maximum concentration that they report in their measured data sets is around 15 mpn per litre. So we only measured less than 15 in about 10% of cases in our study. So you can see that that 15 mpn probably is not representative of all stormwater catchments, particularly this particular site, the Troups Creek catchment. That has quite a significant influence on any quantitative microbial risk assessment that you'll follow, because if you're using a maximum concentration of Campylobacter of 15 mpn per litre to predict risks or to determine treatment requirements for reuse of urban stormwater, you could imagine that if you're using 15 instead of 1,000, that treatment requirement or the risk would be quite significantly higher. So I think Sunny Jian will be talking a little bit about quantitative microbial risk assessments after me. So we thought, okay, maybe it's just in Troups Creek that we're finding high levels of Campylobacter. So we looked into our previous data sets for a few of the other sites around Melbourne, and as you can see here, So if you just click once more, you should have a blue table that kind of comes up on the same page still. See the numbers of Campylobacter in these three catchments are the 95th percentiles are certainly greater than the 15 mpn per litre, and sometimes up to a few hundred per litre. So it's clearly not just one site specific, and this is now, we've done this at another 3 drains now, and we're finding very similar results. So we've got 6 drains in total, 7 drains in total now, finding very high levels of Campylobacter. Why are we interested in Campylobacter? Campylobacter in Melbourne is the #1 leading cause of gastro. So it's a really important microbe to be looking into. I'm not sure if you can see this video, so for those of you who can, then understanding the level of Campylobacter in this wetland was even more evident after our latest field trip. So if you click once more, you'll see a video, I hope. If you can't, then it's a pity, because you'll see people swimming in the wetland that we're monitoring. And those people swimming in the wetland would be exposed to on that day around 500 mpn per litre of Campylobacter. If you put that through a risk assessment model, the probability of infection is rather high. So I'm not sure what happened to these 2 guys, but I would expect they probably had some problems the next day. So we thought, okay, let's have a look into this into a little bit more detail, because we thought, well, are all the Campylobacter that we're finding, although they're culturable, are they actually infectious? So are they actually potentially disease-causing if humans are exposed to them? And so we would love to be able to do another human feeding trial as scientists, but clearly that's not going to happen, so we can't feed these Campylobacters that we find in the environment to humans. That would be not very nice at all. So the second best way is to start looking at, potentially looking at the full genome of these Campylobacter isolates. So that's what we did. We started extracting the DNA and sending them off to sequencing, and we've done it for 200 environmental isolates now, ranging from stormwater, river waters. We've also collected animal feces and sequenced them. We're doing them for human clinical isolates as well, obtained from hospitals around Melbourne. And to begin with, what we've done is we've just extracted these 7 housekeeping genes from these genomes and applied what we call multi-locus sequence typing. And we can then use the types that have been identified using this approach and compare them to databases, international databases, to see what or where these isolates have been obtained from previously. So, for example, we get an isolate from the environment, and we extract these 7 housekeeping genes and find out that it's a sequence type A, and we look at that sequence type on the internet and find that this particular isolate has been identified. And this is just a pie chart. If you click again, you'll see a pie chart appear. That pie chart demonstrates that around 60% of the isolates that are in this international database have actually been obtained from human stool samples and human blood samples, clearly demonstrating that this particular sequence type of Campylobacter has potential to cause human infection. We can do that quite a lot, and we can compare all our isolates that we obtained from the environment to all these international databases. And we can, so if you just click once more, 2 more pie charts will appear. And so we found that about 20% of our isolates that we obtained from these sequences were linked to previous human infection, whilst the other 80% were never isolated from anything to do with a human, and were mainly isolated from waterfowl. So that gives us a better understanding about potentially what level of infectiousness these Campylobacters could be. We're also looking at comparing their full genomes together to see whether or not we can extract some information about how closely they relate to full databases as well, but also extracting things like antibiotic resistant genes and seeing whether or not they're prevalent. Interestingly enough, we found that 2 of our isolates from the environment genetically matched with less than 200 single nucleotide polymorphisms differences in their genomes to our clinical isolates, which suggests significant local relevance. And so I'm quite interested in that, especially considering we've only done about 20 clinical isolates so far in Melbourne. So we didn't expect to find 2 already that had pretty good matches. If you click once more, sorry, the text is kind of hidden by the slide. We wanted to do more than just genetic-based analyses, because we wanted to see whether or not these guys could actually start invading into cells and producing cytotoxins. So what we also are doing what we call attachment invasion assays onto human cell lines to see if these Campylobacters can actually invade into the cells in vitro experiments. We're also doing cytotoxicity tests to see if they can actually produce the toxins capable of damaging human cells. And we're doing some motility tests as well, because motility is a function required for infection for Campylobacter. Well, not required, but very prevalent in infectious Campylobacter. If you click once more, you see a big, ugly green table, just demonstrating that we're not just looking at Campy and bacteria, we're also looking for other pathogens, so looking at adenoviruses, enteroviruses, Giardia, and crypto. I don't want you to go through the numbers there, other than to say that we're finding adenoviruses, enteroviruses, Giardia, and crypto during wet weather events, but not yet during dry weather, although there are very few samples so far. These are all culture-based methods that I'm providing. The other thing that we're doing, so if you click once more, you'll see a lot of pretty colours come up with the titles inflow, outflow, waterfowl, feces, and sediments. The other thing that we're doing at the moment is just doing some more sequencing to identify other pathogens that might be of potential concern in urban stormwater. And so the bar charts that are represented here basically represent the sequencing that we've done on the 16S gene. We're amplifying the V3, the variable regions 3 and 4, and basically using that to identify down to genus level the different bacteria that might be present in urban stormwater, utilising that to help us inform what type of other maybe more specific qPCR tests we can do to identify other pathogens of concern in urban stormwater. I wanted to cover a little bit about microbial source tracking, so I hope I have time. If I don't, then you can just mute me and run, like I did before. But I wanted to present just a little bit about microbial source tracking. So for those of you who don't know what microbial source tracking is, the objective is just to identify where the microbes are actually originating from. And the reason why we want to do that is because we want a better understanding of risks. So it's a common belief that microorganisms that are sourced from human sources, such as failing septic systems or potentially sanitary sewer overflows or combined sewer overflows, would potentially cause a higher level of risk to humans utilising these water sources downstream than the feces of, for example, cows or wildlife, other wildlife animals or agricultural animals. So we're really interested in whether or not we can differentiate between human and non-human sources of contamination. And also from a management perspective, so if we understand where the sources of fecal microorganisms are coming from or originating from, then clearly we can have a better chance of mitigating that risk or mitigating that source of contamination than if we didn't know where they were coming from. So in the early days, we started using chemical markers such as fecal sterols, and this is just a nice little graph demonstrating some of the work that we did, but I'm going to skip that now to the next slide. So the slide now is titled Fecal Sterols versus Caffeine. So we used a lot of chemical markers in the early years to see if we can identify sources of contamination, in particular human sources of fecal contamination into urban stormwater systems. And whilst we found there was fairly good inference of whether or not there was human contamination in there, we did start applying these markers more widely to other catchments, and we fell into a whole heap of problems. One is just the caffeine example. Unfortunately, in Melbourne, in urban areas, there's a whole heap of coffee shops. So if you've been to Melbourne, you know that we're very particular about our coffee. I'm not sure why, but we are. And so there's a lot of coffee shops, and unfortunately they dispose of their coffee grinds into waste bins that often don't have lids. So when it rains, we get this caffeine that comes out into the storm sewer. So what we were finding was that if any of these urban catchments actually had commercial areas in them or a few cafes, we had quite high levels of caffeine detection. So that kind of put that marker out into the bin. If you click once more, you'll have a title called MST Bacteroides. So Bacteroides for the last 10 to 15 years has been a commonly used marker, and the idea is that there's certain strains of Bacteroides that are unique to different animals. So there's one here that I've listed in the first bar chart here. It's called HF183. That's supposedly very specific to humans. Unfortunately, there's a whole heap of evidence that these markers do have cross reactions, and we get a lot of false positives. And in our lab, just as an example, I donated my feces. I thought that was very kind of me. Unfortunately, my feces were analysed for this HF183, and indeed I came up positive, which is good. So it reassured me that I am indeed a human. Unfortunately, though, we also detected HF183 in the cows, in the cow feces that we obtained, which was a bit of a shame. And in fact, according to the cow marker, there's a ruminant marker that's supposed to be specific to ruminants. Unfortunately, I'm also a ruminant. So that's a bit of a problem. So if you click once more, you'll see a little title come up after my picture that says single hit markers. There's a little bit too much room for error. And so there's a little bit of a work in the literature at the moment that suggests that instead of using just one marker for microbial source tracking, we should be utilising multiple markers to make inferences about the sources of fecal contamination. If you click once more, you'll see, again, a title that says MSD Bacterial Community Profiling. And it's got 3 steps, step 1, step 2, and step 3. The idea here is that instead of using one microorganism to infer a source, what we're trying to do is use an entire community fingerprint to determine sources of fecal contamination. So the idea is that we go into our catchment and we collect heaps of source samples. So this is step 1. We go out and collect bird, human, and cow in this example, and we collect them multiple times, and we sequence the 16S gene, and we look at this variable to region 3 and 4, and we identify the fingerprints of the different organisms that could be potentially in each of these sources. The second step is to obtain the water sample that we want to know where the sources of contamination are coming from. And so we call this a sink. So we go out and grab a sample of stormwater, for example. We do the exact same process, and we extract the DNA, and we go to sequencing, and we find out this profile. Then we use some Bayesian modelling. That Bayesian modelling basically just compares the sink profile to all these different source profiles. And it does it in a way which, in the end, provides us with a nice simple estimate of what the proportion is, or the likely proportion is, of contamination between human, bird, and cow, for example, in this simple example. And we can now apply this, and we've demonstrated the application of this process to a whole range of different stormwater systems and river systems around Melbourne, demonstrating its ability to identify sources of contamination. If we were to go back to that Troups Creek example, so click once more, you'll see another blue table that comes up with bat, bird, gull, horse, human, and rabbit. This is using that bacterial community profiling approach on the Troups Creek inlet. And as you can see, the majority of the samples that we've collected here identify human as a significant marker of contamination in this influent, really quite highly contaminated with human feces. Now, these numbers here are taken during dry weather periods, and so they are quite high. We know in this catchment there are leaking septic systems, and so I'm not super surprised by these numbers. I'm probably higher than I expected, but at least we're identifying that maybe some of these Campylobacter are actually indeed human, which is confirmed by some of our sequencing data sets. So if you click once more, you just come to slide 20, which is the conclusions. So fecal indicators in stormwater and MOPUS, we can try to model them. The model's not perfect, but at least it's a first start. Pathogens in monitoring in urban stormwater, they're there. I suppose that's not really of particular interest there to us, but Campylobacter was certainly higher than we expected. But I suppose we have a question about what do they really mean from a human infection perspective. Are they actually infectious? And I think we need to do a lot more work to understand that question. And microbial source tracking in stormwater, can we continue using these one-hit markers, or should we go to this broader, full community scale microbial source tracking, or at least using multiple sources of evidence, multiple lines of evidence to do microbial source tracking? And I think if you click once more, the slideshow is over. So that brings me to an end.
Beautiful, beautiful, David. Thank you very much for that. I think that was a remarkable overview of all the great things that you're doing. You know, one of the things that struck me, and then I want to open it up for broader discussion, was your use of Bayesian statistics to back out the likely partitioning of the sources among the various categories. One of the benefits, I guess, of Bayesian statistics is it also provides some estimate of error. And I'm wondering, those pie charts are obviously they're not, their lines are not fuzzy boundaries, but I would be very interested to know, what the likely error is associated with those numbers as well.
Yep, very good question and good comment. So the errors are huge. So yeah, you're right. I didn't show any of the specifics there, but the errors are quite significant, and the process is quite a complicated process to get this source tracking. It's called source tracker, the actual model, to get it working well. And the problem is that the source tracker model has to do what we call rarefication before it does any of those Bayesian statistics. So we have to rarefy the data to a certain level first. So that means basically randomly sampling that big community to begin with, and only sampling potentially 100 of the sequences that of the 40,000 that we have per sample, for example. And so, in fact, each time you run the model, you may get a different result. And so we've identified that as one of the problems, and we've looked at the sensitivity of all of these different parameters that are involved in the modeling, in the Bayesian modeling, including the rarefaction depth. So if we just rarefy to 1,000 versus 10,000 versus 30,000, what are the results? And indeed, they do change. As you probably would anticipate, the higher the contribution, so if there's a really strong signature from a particular source, those uncertainty bands get reduced quite significantly. For those sources of contamination which are very minute, which could be, for example, human wastewaters in typical urban stormwater systems, maybe not in Troups Creek, but in others, the error bars get quite significant. You're up to the 100% range. So you can, if you're estimating a mean of 5%, then you could be between plus and minus 100% of that number. So they can be quite large, and we think that the degree of that error has a lot to do with the number of samples that we have in our database, which is actually quite low at the moment to derive a community profile, an adequate community profile of a particular source. And also the fact that we're only going down to genus level, and those that's really not specific enough to start getting really accurate information around particular sources of fecal contamination. So there's a whole heap of work to be done. But at the moment, yes, the error bars are quite large. I can give more specific examples, but yeah.
Thank you. That's a fabulous answer. Are there any other questions or comments for David?
Yeah, I had one. This is Brandon Winfrey. I was wondering if you had any thoughts on the best type of green infrastructure, or WSUD, to deal with the pathogens in the stormwater. Which ones are better at not just treatment, but maybe just reducing exposure to pathogens in stormwater?
Okay, good question. I actually had those slides in here today. Unfortunately, I deleted them, so maybe I should put them back on. I don't know. Anyway, so I had 10 slides, but it was just too long. So we have monitored a range of LID or WSUD, or whatever you want to call them, green infrastructure systems for pathogen removal. The one that we've been focused on a lot at Monash has been biofiltration systems, and indeed we've had a good collaboration with UCI on doing some work on virus removal in these biofiltration systems. I believe that Anna mentioned the fact that we're trying to incorporate copper-coated zeolites in these biofilters as well, which can also help improve removal of pathogens through these systems. Indeed, we do get 1 to 2 log reductions of most pathogens. So we've monitored these systems for actual pathogens, not just indicator organisms, in optimized conditions. So depending on wet and drying regimes and the optimal design of the biofiltration systems, I think you could push, I think we aim to push them to 2.4 log reduction for bacteria, 1.2 for protozoa, and 2.2 for viruses. And we succeeded with that when we incorporated the copper-coated zeolites into the media. But we're trying a whole heap of other ideas, like using antimicrobial plants so plants that excrete antimicrobial compounds into the soils to help reduce pathogen levels. Dosing these systems with different microorganism communities to see if we can kind of outcompete the pathogens that are introduced into the system. We want to get around introducing copper into a natural biofiltration system. We've done a lot of monitoring of wetlands. To be honest, I don't believe in wetlands. The wetland that I was speaking about, the Troups Creek wetland, typically produces Campylobacter. We're doing some source tracking which identifies it's probably coming from waterfowl that live within the wetland itself. We're not getting much inactivation, if any at all, through the wetland via irradiation or even settling. So wetlands we're not seeing as very promising technologies at the moment, but biofiltration systems certainly look like a goer.
Great. Any other questions for David?
I have a question if you can hear me.
Absolutely. Go ahead, Steady.
Okay. Yeah. Hey, David. Well, I missed most of your slide as your talk because I couldn't load them. Later on, I got the downloaded a smaller version of your slide. I'm looking at you have a pathogen in urban water, urban stormwater. There is a table you presented, and there is HPV as a marker. There's a very high rate of positive detection.
Yeah, human polymer. Yep.
Is that human papillomaviruses in the stormwater? Is that a marker of human sewage?
Yes, that's what they were used. So this is Simon Tozer's group in CSIRO utilizing that. So that's on slide 7 for those of you who got that up.
And then all through the
very high level.
sorry. Go ahead. I didn't catch.
No, I was just saying that it's a very high level of detection, like we're talking.
Right. Then you just continue of working on it. Why is that?
Sorry, say that again.
The data for HPV have very high detection level, and then there is most of the Victoria and the West Australia sample was not testing for HPV. Is that correct?
Yeah, that's correct. Yeah, so we didn't test for them at all in those samples.
So those are by PCR method?
All PCR. Yeah, we had a lot of. When I say we, I'm talking about our collaborators at CSIRO. Had a lot of problems with inhibition, especially with the Victorian samples.
So by comparing those sort of stormwater from all different states of Australia, do you see a common trend and pattern from this?
I personally don't. I don't. My personal opinion is that I prefer to utilize culture-based data, and so that's why I skipped over this as quickly as I could. Because my personal belief is that we should be looking at culture information. I know that you can't do that for a lot of the viruses. So there is a report on this that I can send through. Actually, I could probably make available on maybe the PIRE website or maybe this Google form. I'm not sure how to make that happen. So the guys at CSIRO have created a report on this and looked into patterns, temporal and spatial patterns of all of these indicators or pathogens that they've been looking at. So yeah, I think the report is probably your best bet to get more information about it.
And also to understand the risk and not only seeing positive or negative for pathogen, the other very critical information is the dose, is the concentration. And then we have been struggling trying to figure out how to convert those positive detection into certain type of concentration so that we have a better understanding of the risk. If I do look at your report with the concentration information on it, which is slide number 12, and most of the detection are below detection limit.
Which slide are you talking about, sorry?
Number 12. I think
number
this
12.
is 12.
12. Ah, here we go. Yes. Okay. Yeah, definitely. And we're talking like quite large volumes here. So we're not talking small volumes of water done. So for the crypto Giardia, it's often more than. Sometimes we can only get 10 litres, depending on the size of the event. But often there's more than 30 litres of stormwater that's being filtered.
Is this also PCR-based method?
No. So the slide 12 is all, well, not all of it's culture. So the Giardia crypto is done by the US 1623 23. Sorry, I forget numbers nowadays. US 1623 standard for geodocrypto microscopic observations, but we also do a cell line assay for crypto to see if there's any viable crypto there. Adenoviruses and enteroviruses using the A549 cell line to enumerate adenoviruses and enteroviruses using the 11 NPN method.
Right, interesting. The only positive results coming from the adenoviruses in here.
Yeah. It's we have more data than this. This is just for one site, so this Troups Creek wetland. We have other sites or other urban creeks around Melbourne where we've got more data than this.
Very interesting. I think those will be a very valuable data set and for I guess the following up the risk quantification. So thank you.
Yeah. What do you think about the Campylobacter, Sonny? Is this okay for me to ask a question, Stan?
Absolutely. Go right ahead.
Well, the number is quite high, but I think maybe we need to understand a little bit more of those Campylobacter do have the sort of disease-causing capability or not. I think
my
we don't
hypothesis
know
is
the
that it's much lower than. There's not a lot of good information around on that proportion of infectious. It's very sporadic, and people say, 20% or something, and just apply a fixed number of 20%, which is odd. But yeah, anyway, it's interesting.
Well, that's great. Maybe in the interest of time, we could transition to the next talk. So thank you very much, David. And next up is Sunny.